All right, well, good evening, everyone. Good evening, everyone. There we go. That's a little better. Maybe I'll put this on my tie instead. We are almost there, guys. Like, one more test. Do any of you had, like, 2 more exams? Yeah? Okay. All right. For me, it's just 1. Okay. All right. Well, we're almost there, guys. Summer is around the corner. I'm looking forward to golfing, that's for sure. Just bought my membership. You probably could have figured out the guy that wears suits, he likes golfing or whatever. Okay. I guess first I want to update you on a few of the things that have been happening kind of beside our lectures throughout the year. First, the Senate. So many of you probably voted for me for Senate. Thanks again for that. My term starts this coming month. And so I'll be sending out updates about the Senate meetings. That was one of the main parts of my campaign, was keeping the people on the ground, that is you guys, informed about what goes on at the upper levels of government at UW. So those updates will be coming out monthly. There are 2 months in the summer where meetings don't happen, but through the school year, it'll be roughly monthly that you'll hear these updates. One of the main things, I don't know if you've heard about this yet, our university is in a little bit of financial trouble. We're projected for a $75 million deficit for this coming year. The Senate has approved pay cut measures, a few other things as well, to control the deficit, but essentially it's not sustainable. That's the thing to remember. We're having to draw from our central reserves. So once those central reserves run out, we're kind of in a problem. Now, some of this is due to provincial funding and a few other things, but basically there's going to be a little bit less hiring happening here, maybe a little bit less expansion, but hopefully we're able to right the ship in the coming years, and I'll look forward to keeping you updated about that. Now, at the last review lecture, I asked you guys to participate in a science super vote, okay? Now, the last WUSA general meeting, it was actually kind of crazy, the amount of people who showed up. Much more than last year. So the super vote that we had was exactly counteracted by another person, get this, who had the exact same idea as me and got just as many proxy votes from other students. So unfortunately, the results of that meeting is that we approved more increases to fees for students. Unfortunately. Now they're not huge, so like individually, you know, you're maybe paying like $5 more, something like that, right? Compounding though over years, you can see how that might be concerning, right? So yeah, in the future, if you guys want to show up to WUSA meetings, you can. I gotta say, there's a lot of drama at this last one. One of the executives who was elected in the last WUSA election actually got removed from the slate of directors that was approved. So they actually removed him off because of misconduct in another election. And it's actually a long story, and it was a very dramatic meeting, lots of shouting, this kind of stuff. So maybe there's a recording of it somewhere. I don't know. It was actually kind of wild, though. So I guess you missed out on that, but whatever. Okay, so that's kind of the update on the WUSA politics, the university politics. Now I guess we're getting into what you all came here for. There's a lot of people here today, so thank you. Hopefully there's enough seats for everyone. Maybe you have to get people to split up. So the way I've structured our lecture today is not so much me talking at you about all the content, because first of all, there's way too much content to do that. And second, the active recall is really what we need to lean on here for studying most effectively. So what we're gonna do today, I have 20-ish, 21 questions that I'm gonna ask you. Some of them we can get through quite quickly. Other ones are gonna take a little bit of time. For reference, the longest one is going to be a full page solution of work. So what I want you to do now is, if you haven't already, get out a piece of paper or a binder. If you use an iPad or a computer, get that out. Get out a calculator, you're gonna be needing it later. And the other thing I want you to do is find people around you that you know or are willing to get to know. I'm thinking a partner or at max 3 people, okay? Yeah. Okay, so hopefully you've got your partner, and the idea with this is that for some of the questions that require you to discuss the answer, you're going to discuss with your partners. Okay, other times you're just going to be doing work like this, so maybe less talking for that. The room just descended into anarchy there for a bit. Wow, okay. Man, what an experience. All right, guys, we need to get it together so we can get into this. The hope for this lecture is to be an hour and a half long. Now, I have no idea if it's going to be, a little bit shorter or longer on that side. We're going to try to shoot for an hour and a half. I can also just stop the questions after an hour and a half. That could work too. So, again, you're welcome to leave whenever you need to as well. But, hopefully you benefit from the questions. Key thing that I always clarify at lectures now, this was a confusion at the first lecture, I am definitely not a TA, okay? I am taking this test. I am just as worried about this exam as you are, okay? I know that seems weird, but literally, there's some anxiety going on here if you can't see it, okay? So, I'm not the expert, but what I'm hoping to do is facilitate group work, okay? We can all learn from each other. That's the point of this lecture, not learning from Jordan. Okay, if you want to keep up with future lectures, I guess there's no more this year. Maybe next year I'll keep doing some, who knows. That's my Instagram there. That's also where you're going to get Senate updates. Okay, now we're going to get into this. It's roughly equal distribution between all of the units. I know the exam is not like that, so frankly, my apologies. But I have prepared more questions from previous lectures, so that's why I'm kind of doing this evenly. Feel free in your own study time to put more of an emphasis on those later slide decks. So to start, this is an easy question. Okay, what is the definition of a species, the proper definition of a species? And what are two types of selection that are responsible for the evolution of species? You've got like 30 seconds, and then go. If you can think of examples of these, try to think of an example of the two types of selection, one of each. Okay, guys, we're going to get into this. Hopefully you've put down some answers there. Okay, so what is a species? Okay, the first thing to recognize, this is like the basic definition if you look it up on Wikipedia, okay? If you've got a population that can inbreed, that is breed within the population and produce fertile offspring, then you've got yourself a species, okay? An example of a case that's not a species, right, is a horse and I believe a donkey, right? And they make a mule. But the mule, right, is not fertile, okay? So that's not like a species, right? That's the key thing to remember there. The other thing to remember is that they share the same genes. A species shares the same genes. They have different alleles, right? Hence we have variation within the species. But to be part of the same species, they've got to have the same genes, the same loci on the chromosomes. Now, hopefully you'll have remembered natural selection and artificial selection. Should be very central to genetics. Artificial selection, this is an example we talked about in the first lecture. Humans can select for certain traits, okay? And this is not in a Mendelian sense where you look at all the genotypes and try to figure out the ratios. This is simply saying, let's look at a population, for example, foxes, select for a trait, in this example, tameness, and then breed the most tame members of one generation together to produce what's hopefully a more tame second generation. Take the most tame from that generation and keep breeding. So then you get from like a fox, right, down to like what is almost considered a dog, right? And there's domestication syndrome which goes with this. You can see the difference in physical characteristics there. Okay, natural selection. So this is not a human agent making the decisions. This is environmental pressures shaping what alleles are selected for. And it's really just utility within the environment, right? A white rabbit will blend in well to the snow behind it. Hence, it can avoid predators more, it will be killed less, and it will be able to pass on its genes, that is, its alleles for white fur, to more offspring compared to other rabbits that don't have a white coat. That's the idea of natural selection. Okay, another more simple question. Define the laws of independent assortment and segregation. Again, like 30 seconds for this. Okay, all right, so the laws of independent assortment and segregation. Okay, segregation. The idea here is that you're looking at one gene specifically. What happens for a certain gene when we transmit alleles from one generation to the next, okay? And this is the idea that every organism, every diploid organism, has two alleles, right? And those alleles will separate from each other, and one will be passed on to the progeny at random. Okay, it's a random process of deciding which allele is passed on. This is, again, I stress, within an individual gene, right? For every gene, the two alleles separate, one is passed on at random. Okay, now the law of independent assortment, this applies to multiple genes within the organism. How do they interact, okay? And so the idea with this is that the inheritance at one gene does not affect what happens at another gene, right? If I inherit a certain allele over here on a certain chromosome, right, on another chromosome, which allele is going to be inherited, it's completely random. They have no relationship between each other, okay? There's one instance that we have covered in this course where the law of independent assortment has been violated. And what case would that be? Can someone just quickly tell me that? Yes? Yes, and that would be an example of... Yeah, that's the word I'm looking for, but that's the right concept, right? The idea that they travel together on the same chromosome. And the discovery of chromosomes as the unit of inheritance that Mendel referenced through his statistical analysis is something that's been really central to the progression of genetics, right? The fact that Mendel was able to predict that there were things called chromosomes before we ever knew that they were there, that's pretty insane, right? And that speaks to the incredible utility of this theory, right? The incredible ability to predict. Okay, next question, again, another pretty cheap one. I want you to explain what each of these are, like 30 seconds max. Okay, everyone. Either you got it or you didn't. So, actually, do I want to have like a volunteer, just someone who's like really confident to say, hey, I know exactly what all these are? Yes. Yep.
The one on the right is partial penetrance and complete full expressivity.
Yep.
The one on the top is varying expressivity but full penetrance. The one on the bottom is varying expressivity and varying penetrance.
Yep, that's right. That's right. These are like different terms for that, but it's the same idea, right? Constant expression, variable expression, incomplete penetrance, and then a combination of both incomplete penetrance and variable expression. Examples of penetrance: retinoblastoma. That's one we covered recently, right? Another one would be the BRCA genes, right, for cancer. Those have a high degree of penetrance relative to what cancers are like. It's about 44%. You can see that on the slides. So that's where these concepts are actually relevant in the world. Okay, now guys, I want you to prepare yourself for this. Get out a piece of paper for sure here. This is a kind of a brutal question, all right? This is the upper echelon of problem you could get with like ratios of inheritance stuff, okay? The first time I tried this, I like kind of got it right, but kind of not, okay? So I'm gonna give a set amount of time. We're gonna say maybe 3 or 4 minutes. At the end of that time, we're just gonna take it up, regardless if you got it right or not. Okay. Yeah, if you're over on that side, you can like take a picture of it or like lean over or whatever. Yeah. This is not a problem that I made up myself. This is a problem that I took from an old edition of the textbook. So this is not some brainchild of myself. This is a perfectly legitimate question from the textbook with a perfectly legitimate answer as well. Okay, how's it coming along, guys? Like, how much more do we need here? Are we good? Does anyone not want me to move on? Okay. All right, we're gonna keep going then. Okay, let me flip to the answer here. All right, so here's the idea. You can start reading this, but the idea is that in the F2 generation, okay, we have 4 different phenotypes. 4 different phenotypes. We know that one gene, one locus, cannot produce 4 different phenotypes. It can produce 3, but not 4. Okay. Now, in terms of modes of inheritance, right? We have like epistasis, right? But epistasis, we have 3 different phenotypes, right? Whether that's dominant or recessive. You can have complementary gene action. That'd be like a 9-to-7 ratio. That'd be 2 types. So again, doesn't fit. What you end up coming to is that the two genes contribute like a dihybrid cross, right? Where you have the 9-to-3 to 3-to-1 ratio. That's basically what this is. But they converge on one trait. So instead of determining 2 traits, they determine 1. That is coat color, okay? So the first hint we're going to take is that when you cross the black and the chestnut at the top, right, you get a different colored offspring. You get a different colored offspring. So that means that one of the parental types is not dominant in both genes, okay? So one is dominant in one gene, one is dominant in the other. And so what you can come up with is this idea here on the next slide, that black is dominant at one, and that chestnut is dominant at the other. Now, why are they pure breeding, right? Why are they homozygous? Because they all produce one type of offspring. There's no variation in color for the F1 generation. It's all bay, right? And so that's how we can get the production of a third coat color in the F1. Okay, then, like any dihybrid, right, that has 2 genes, we get 4 different classes of offspring, all of these. Now, the reason that they mention liver, right, is to help you understand that the liver is the homozygous recessive at both genes. When you cross it with the black parent, right, you get the black phenotype. So there's no contribution from the liver there, right? When you cross it with the chestnut, it also has no contribution. You get a chestnut again. So the idea with this is that liver is unable to contribute, thus you can assume that it is homozygous at both genes. Okay, that's the logic there. I'll be posting these slides afterwards for you to take a look. But again, keep in mind, this is something that was covered at the end of the slideshow for, the relevant slideshow for this. And it's one of the many examples you could see. Okay, it's also one of the harder ones. So, take heart. Okay, here's the next one. I made this question up, all right? I do have a brother and sister-in-law. Okay, they don't have 4 kids, they have 3. But I added another kid just for the fun of it. Okay, Jordan's brother and sister-in-law have 4 kids, 3 kids, okay, 4. Jordan's brother has brown hair, like myself, and is heterozygous, okay? Jordan's sister-in-law has blonde hair and is homozygous recessive. Blonde, just generally a recessive trait. What is the probability that 3 out of 4 kids have blonde hair? This has been like genetics before my eyes, seriously. Like, after every kid is born, I'm like, brown hair, blonde hair. I don't know, right? It's a relevant part of my life. That's for your help. You probably knew how to draw that already, but there it is. Hopefully you're actively computing this, if not already done. I want every person to have an answer down on their sheet. That's what makes this review good.
People are probably not.
Yeah, I mean, you still need to make calculations like this. It's like A, B, or C. Give it a go. Okay, how are we coming, guys? Do you need more time, or are you done?
We're good, Jordan.
You good? Okay. Okay. All right, guys. We're gonna see the answer here. Okay. The first thing that you should be realizing here is taking a look at the genotypes and asking yourself, for any given niece or nephew that Jordan has, the probability of them having brown or blonde hair is 50%, right? 0.5. We all got that far? Okay, good. All right, all right. So now, hopefully, you recognize you need to use Pascal's triangle, right? You also recognize that there are 4 kids, so you need to look at the n equals 4 line on Pascal's triangle, right? Okay. So what is the expression, what part of that binomial expansion do we pull out to calculate this, right? Okay, well, we look for the one that says p to the 3, q to the 1, right? And we let p and q be equal to the probabilities of getting brown hair or blonde hair. So I'm going to flip to this slide, which says that. You can see that p and q, 1 for the probability of getting blonde, 1 for the probability of getting brown, okay? And we pull out that expansion, you see 4p to the 3, q1, right? And we count that by going like this. P to the 4 is this one over here. P to the 3, P to the 2, P to the 1, P to the 0. And then for Q, Q to the 0, Q to the 1, Q to the 2, Q to the 3, Q to the 4, right? That's how a full expansion works. And this is the coefficient that you put beside it. So if I'm going to write the coefficient, it's going to be that. You see that right there? Okay, all right. Then we plug in our values for P and Q. And the probability that you're going to get is 0.25. Is that what you got or not?
Yes, Jordan.
Yes, good. I made that question up, so I'm glad it's right. Come on. Not pulling a chem prof on this one. Laura's great. Everyone else is not. Laura's great. I'm kidding. I don't know about the other guy. It's just, ugh. Okay. Hey, I'm glad that's over. I'm just going to put that out there. I'm glad we've written that exam. Okay, guys. Next question. Okay. Autosomal or sex-linked? You figure it out. Yes. I can't hear you. What? Oh, just think about it. Okay, guys, I'm gonna bring it back. Okay. So, in this case, if you're unsure, you should really go for the explanation that has like the most going for it, the most explanatory power for it. Okay. The first thing that you should recognize, and I think I actually got this answer wrong. Like the first time I looked at this, I tried to do it in like 15 seconds. But think about this: there are more males affected than females by like a fairly significant margin. It could be due to error, but look at that, right? There are more males affected than females. Sex linked, right? Sex linked. The other thing that you should recognize, see this one in the middle here. I'm going to flip to the next slide so you can see it there. The one in the middle, right? That one, you've got a female that's affected, but a male that's unaffected, right? So both males are affected. That's very consistent with sex linked, right? And no females are affected. Again, it's not like 100% this is it, but it's like it's really good proof, right? That this is what's going on. And again, what you'd be seeing, right, is that the male offspring, they get the Y from the unaffected father, and they get the X from the affected mother, right? But the daughters of that cross, they get an X from their father, right? And if this is a recessive condition, then they won't be affected. So that's what you should be looking at for this. Does that make sense? Yes, we're good with that? All right. Next question. This is the second one and the last one we're going to be doing on this. Tell me what the pattern of inheritance is, just straight up. What is it? All right, have we all got a hypothesis? We good to go? Okay, we're set. All right. Does anyone want to put out a hypothesis, what they think it is? Yes, it's Steve in the back. Yes. Yes. Okay, let's think about why, all right? Okay, so the first piece of evidence you want to look at is this second generation going down to the third. Okay, how is it that we got an affected offspring without either of the two parents being visibly affected, right? Well, not dominant, right? It's got to be recessive. The recessive is how you can mask that. All right, now the other place you want to look is the top right, like generation 1 over to the right there. Okay, if this is sex-linked recessive, then you would expect all of the, like that male right there to be affected, right? Because if the mother's affected, then she has to pass on one of those X's to the son, right? Because he gets his Y from the father, but he's not affected. So it's got to be autosomal recessive. There's exactly what I said right there. Does that make sense? Does that line up with what you said? Okay, good. Next question. Okay. How is sex determined in Drosophila? And then kind of on top of this, is an XXY Drosophila a male or female? Okay, let's pull it back. I know this was a question on the test, and I bet some people got it wrong. It may show up on our exam because I'm sure it was a sticking point. Okay, does someone want to volunteer an answer? How is sex determined in Drosophila? Yes. That's what you might think. It's actually not the case. 2 X chromosomes does not make it a female. Yes.
Is it the X chromosome ratio to the autosome ratio?
Yes. There we go. Okay. So it's the same idea because usually the Drosophila is diploid, right? So if you have 2 X chromosomes and then 2 sets of autosomes, a 1-to-1 ratio indicates female, and then a 2-to-1 ratio that is like 1 X to 2 sets of autosomes, that's a male, right? So in this case, what is that there? That would be like a female, right? Because it has 2 X's. The Y doesn't matter. In humans, we have an SRY region on our Y chromosome, okay? And that like codes maleness, essentially. And it's really the only thing on the Y chromosome that's like useful, I think, from my reading. And the point with that is that any human that has a Y chromosome will show male characteristics. But for Drosophila, that's not the case. There's no gene on the Y chromosome that makes it male, right? It's the ratio of X chromosomes. Now, there are a bunch of other organisms. By the way, there's the table for that. There's a bunch of other organisms that have different ways of sex determination. Some are temperature dependent, right? That's kind of wild. And there are other ones that are actually the reverse of humans. So instead of females being XX and males being XY, the males are now ZZ, and the females are ZW, okay? So the heterogametic and the homogametic, they're reversed. Okay, does that make sense? Keep that in mind, okay? If you didn't know that, just have that in the back of your brain somewhere. That may come up on the exam. All right, are we all good with that?
Yes.
Yes, solid. Thanks, John. Appreciate that, man. Okay, now the question. Compare and contrast oogenesis and spermatogenesis. And here's the kicker of a question B. What are the 4 mechanisms of parthenogenesis? And if you can relate this to Komodo dragons, you're doing really well. I saw you on when I was driving in. Yeah. You have a card? Would you like to hang out somewhere else before coming to campus? I see. Okay. All right, everyone. We're gonna bring it back. Hopefully, if anything, you got A, right? That's what we're hoping for. If you got like 1 or 2 for B, you're doing pretty good. Okay. So we're gonna start with A. Oogenesis there on the right. Okay. The key thing to remember here is that you're only producing 1 gamete from the process of meiosis. These are both processes of meiosis. Again, one for the females, one for males, right? But only 1 is viable. The other 3 products of the meiosis process, the first polar body and then the 2 second polar bodies, they're not used. They're eventually degraded, right? Only 1 egg is produced from that process. For sperm, on the other hand, you get 4, right? 4 equal products. And eventually they develop into sperm. So it's the spermatid and then down to sperm. You need to add the flagellum and various other components. Okay. Now, parthenogenic eggs. Okay. This is a little bit wild. So the Komodo dragon reference is in the slides, okay? And basically, they are the reverse of humans, right? They have the ZZ and the ZW. So the only viable offspring are going to be males, because through these for all of these processes, the sex has to be homogametic. So let's just go through each one of these here. The first one, you've got your germ cell at the top there, okay? And it just does mitosis, not meiosis. It's like, meiosis, here's my path. Nope, other direction, right? Like complete sight, right? Okay? So at the end, you just get like a regular cell, okay? Just straight from the germ cell. There's no like meiosis process going there whatsoever. Okay, and why there's two there is because it's meiosis, right? You get two products from one initial cell. So one of those would be an egg. And it would be an exact clone. Remember that, right? An exact clone, because that's the process of mitosis, producing an exact copy of that parental cell. Okay, the second method, all right, is we go to meiosis 1, and then you just have a failure of meiosis 2, okay? So you don't need fertilization then, right? You don't need a sperm to come along and fertilize the egg. You already have two sets of autosomes, okay? But they're identical. That's the key thing to remember, right? Humans, we have 46 chromosomes, two sets of 23, right? But imagine those two sets would be identical. They would be exact copies of each other, which is not great in terms of redundancies, right? That means you're like homozygous everything. You're not heterozygous for anything, which, you know, could be problematic in some cases, right? Okay, the third case here is you go all the way through meiosis 1, meiosis 2, but then you just fuse the polar bodies, okay? So one just gets fused to another, and that is in place of fertilization, right? So instead of getting a sperm and an egg, you just get two eggs. Boom, there you go, okay? And that could produce some wild outcomes there. Okay, and then finally, you can go all the way through the end of meiosis, and then instead of being fertilized or having two eggs coming together, instead you just duplicate the chromosomal material, okay? So again, that is going to result in having like two sets of identical autosomes, right? And sex chromosomes too. Everything is identical, so you're homozygous for everything in these cases, okay? The point of this discussion is not so much that you know all of these. It's cool to know all of these. But it's more so that you can remember what goes on in meiosis, right? What intuitively is happening here? What sets come from where, right? All these sorts of things. You're probably going to get a question that says something like, you know, what happens if I like disrupt some part of meiosis 1 or meiosis 2? You know, maybe some Klinefelter syndrome. How can I produce that from non-disjunctions, right? So just having that level of familiarity with what's going on is what's needed. Okay, enough of that. This is a little bit of a long question, okay? I'm going to be honest here. Now, there is no solution to this, so you are the solution. I came up with the solution, of course, but compare yours to mine, and we're going to see if we're right as a room. Get out a piece of paper, okay? Like, it's going to take some time. Like a full sheet of paper, okay? I'm going to time this for about 10 minutes, and at the end of 10 minutes, we'll just walk through the answer. Go ahead. I understand this is basically doing your Drosophila assignment, part A, I think, or not part A, part B? Yeah, part B, yeah, okay. But that was the easiest one, so, you know, hopefully. I urge you all to try to solve this. We have enough time, so please give it a go. I'm going to help you guys out with a little place to start and show you like the first part of my solution. The key thing to do here is to assign alleles, assign symbols. Okay, so you see how we're assigning a letter, right? Like a W, an E, or a B, right? And if there's nothing there, that means it's wild type, right? And if there's something there, then it means it's not wild type. So just based on that, you can write down the letters, right? Wild type or mutant. And then from there, you can put the numbers beside and identify the double crossovers, identify the parental types, then find the map distances, right? Remembering to count the double crossovers twice for the 2 genes that are on the outside. If you don't have a piece of paper, work with the person beside you. Hopefully you've made allele assignments by now. You can see here I pointed out the parental and the double crossovers. From that, at least you can figure out which one is in the middle, right? As like a simple step, if you're not doing something right now, I dare you, just write on a piece of paper which one is in the middle. Just you can put that down. Okay, just for reference, when you're looking at this, the one on the left here, this is basically just a copy of this, but written out nicely. Each of the genotypes are there, each of the frequencies are there, pointed out, and a conclusion about which one is in the middle. So reference your answer there, and then keep going. Keep calculating the actual distances between them, right? The recombination frequency. That's what you're trying to calculate. Then from that, convert that to map units, and then keep going. All right, so for the sake of time, we're going to give it like 2 more minutes, something like that, so you just wrap up. If you are close, that's okay, you don't have to be done. Just make sure you know what's going on and keep going. Okay, guys. that hasn't been 2 minutes, it's been 1 minute. But with where engagement is at, I think we should keep going. And if you're kind of partially through, then you can just see if you're right or not. Okay, so I'll start walking through this, all right? So the first thing to do, of course, is to assign like genotypes, to assign symbols, right? So you can see from up there that if it says wild type at the top, then of course you write 3 symbols, you can assign them whatever, right? I've assigned them here as being W, E, and B, right? W for wing, E for eye, B for body, right? A plus superscript means it's wild type, right? No plus superscript means it is mutant. So whatever mutation that is described on this screen, if there's a mutation there, then I don't put a plus, okay? And then of course I've written them out like this. So now you can see the 2 parental types, right? That is simply the ones with the highest frequency. That's really simple to find, right? And then the 2 with the lowest frequencies are the double crossovers. It's that simple. Okay, so now we need to find the locus, the gene that's in the middle. It's between these 2. So how on earth do we do that? Well, it's pretty simple again. We just compare one of the double crossover types, doesn't matter which one, to one of the parental types. Again, doesn't matter which one. Here I've said W plus, E plus, so 2 wild types and 1 mutant at B, right? Compare that with W plus, E, and B, okay? So this right here is a double crossover type, okay? And we can see that the W plus is the same as the W plus here, right? We can also see that the B is the same as the B here. What is the odd one out? It's E, right? E is the one that's the odd one out. And if you did that with any of the other ones, any other double crossover genotype with any other parental genotype, you would get this result. Okay, then what you're going to do is calculate the map distances. All right, so you can see here W to E. And it's generally good procedure to start with ones that are not on the outside, right? Start with the ones that are middle to outside, and then make your last one the outside. Okay, so we go to W to E. We've got to find genotypes that don't match, that are recombinants compared to the parental, right? So we use the parentals as our reference. Either, so for W and E, either W is mutant and E is wild type, or W is wild type and E is mutant. Okay, so we look for anything that's not that pattern. So anything that's like homozygous, okay? And so when you do that, you add these up here, okay? That's the addition you get, and you get 0.337, okay? And of course, you're dividing by the total number, which we found down here, okay? So that's 33.7 map units. Did anyone get that far? Yes, and you got that. There we go, okay. All right, it's good, because, hey, I did this word too. There was no solution to this, so it's good we're checking with this. Okay, all right, E to B, you go through the same process. You look for recombinant types, okay? And then you run that, and you get 1.4 map units. Wonderful. Okay, then, finally, you've got to find all the recombinants for the outside, okay? Now, when you look through this list, there are a few that are going to be apparent, okay? Namely, the ones that correspond to, like, these here, right? So let's take a look at the parental, right? So the parental, again, is either, so W to B, either 1 wild type and 1 mutant. That's what it is, right? So either W is mutant and B is wild type, or B is mutant and W is wild type, okay? So either or, right? And then you look through the rest of these, you can see these are both wild types, so we've got to count that. These are both wild types, so we've got to count that. These look fine, right? And these look fine. Okay. These are homozygous, so you got to count that. And of course, you have to count this one too. But what about those ones in the middle, right? The 2 and the 1. Those are double crossovers. So we got to count those because there's 2 crossovers that happens. We got to count them twice. So hence you can see this calculation right here, okay, where we've added all them up and we've counted our double crossovers twice. At the end of that, you get 35.0, okay? And you have error plus or minus 1 of the 0.1, okay? That, again, has to do with rounding. Now you can construct a map, right? Because you know which one is in the middle, you know which ones are on the outside, you know the distances between them, so you end up getting this little map here on the bottom, okay? That's essentially what you would want to get. Does everybody track with that? Does everybody get that? Okay, is that what someone got? Did someone get that? Yes, let's go. Okay, all right, that was a success. Okay, next question, guys. We're moving on. Identify the open reading frame and write out the corresponding polypeptide sequence. This is question 11. I've thrown a little wrench in the puzzle as well, you may notice. You may remember open reading frame corresponds to where translation starts and ends, right? There's got to be a start and a stop. Sorry? That is the coding strand. Yes. Coding strand to clarify. Coding strand. Huh? That's what it is. Yeah, the coding strand is the mRNA-like strand. That's right. That's right. This is essentially like I came up with this, but it's one of our tutorial questions. What is the start codon? There's something to think about. What is a start codon? Someone want to put their hand up and tell me what the start codon would be? Yes. ATG, that's right. Not AUG, because this is DNA, not mRNA, right? You can see that from all the T's there. There's no U's in there. If there is, that's a mistake. What are stop codons? There's another one to think about. What are the 3 possible stop codons? Give that a think. It may be helpful to copy this down on a piece of paper and write out sets. 3 triplets, codons. That's it. Both sides of the room. You can see it better. There we go. Okay, everyone. I think are gonna keep going here. All right, let's get going here. Okay. First thing to notice, there are in fact 2 ATGs here, right? So which one do you pick? Yes, the second one. Why is that?
Because it has to go by 3, and the first one is separate.
Okay. What about stop codons? Have you? Yeah. Yes. There you go. Right. There's no stop codon. Now, in our tutorial book, this is an issue I had with it. There was no 3' there. So you said, oh, like what if it just kept going and then there's a stop codon later, right? But that's the 3' end. There has to be a start codon and a stop codon. And without both of those, you don't have an open reading frame. Okay. So you need to have both. And if you see the solution here, our top one, let's see if I can move this over. Man, I should get a pointer. Is this a pointer? Whatever. Okay, there we go. This top one here, you can see that there's just no stop codon. If you go down to the next one, then you can see that there is a stop codon, okay? And if you pull that out, then you get those amino acids crossing with the chart, okay? Do we understand that? Did people get that? Yes? Are there any questions about that? Good. Okay, we're going to keep going. Question 12. This is an easier question, I would say. You kind of know it or you don't, okay? How can bacteria have larger genomes than humans, and yet humans are much more complex? Describe in detail the mechanism, the actual cellular workings that make this possible. In the slides, there's actually a picture. There's like a paramecium, and it has more base pairs than a human. Like, what's up with that? Think about that. I'm going to give you a hint. Introns, exons. Introns, extrons. And then, kind of for part 2, what is the mechanism of dealing with introns and exons? What is the actual machinery that works with those? What's the process? All right, folks. We're gonna keep going. I'm sorry, guys. Technically we have 20 minutes left, so we should keep going here. Okay. The key to this question, guys. Is alternative splicing. Alternative splicing. Okay. So the idea is that for any given section of mRNA, right? You can splice that differently. I should have said that differently. You got a gene, right? And you can have different exons and introns for a given type of mRNA that you're trying to create, right? You can splice out different things from different places. So for any given strand of DNA, there are a bunch of different combinations that I can make and a bunch of different types of proteins. That's the idea with this. Now, how does alternative splicing work? Well, splicing in general, how does that work, right? Spliceosomes and SNRPs are what should be coming to mind here, right? Okay, so here's the basic idea. You've got the 5' side and the 3' side of the intron. The intron is what's taken out, right? The exon, X stands for express, not exit. Express, so we want to express the exon. We get rid of the intron, even though it's in. Just remember it's the opposite, okay? All right, so we make a cut at the 5' side first. You can see that right there, okay? There is a cut there. Okay, now that strand folds over and binds at an A, right? An adenine. From there, you make a cut on the other side, and again, what's making these cuts? The spliceosome, right? The SNRPs of the spliceosome. Then you're able to cut all of those out, and then of course you just ligate the backbone together, and boom, now you have a process of making a mature RNA. Something to think about is what other steps are necessary to make a mature mRNA? What other modifications do you need to make, right? And maybe about the actual physical nature of the cell, what is it that makes this necessary, right? Why do we need to have all of these changes other than just having a larger amount of DNA to draw from, but also what conditions of the cell, right? Would that break down rather easily, right? Do we need it to survive longer? What kind of end things do we need to put on there? Just to have in your mind. Okay. What property of prokaryotes allows them to have polycistronic mRNA? Why can't eukaryotes have polycistronic mRNA? And can you think of a compensation mechanism for this? Question 13. first try defining what polycistronic means. It relates to the lac operon. All right, guys, we're going to think about the solution now. Okay, first of all, what does polycistronic mean? Can someone define that for me? Anyone? Yes. Transcription and translation at the same time. No, that's not the case. What does polycistronic mean? That would be happening in prokaryotes and not eukaryotes, so yes, but that's not what's referred to as polycistronic. Yes?
That can produce more than one protein?
Yeah. So the idea is that you've got an mRNA, right? And you can do multiple rounds of translation on that. Not multiple rounds as in like multiple ribosomes moving through it, but there are different proteins on that one strand of mRNA. Like you've got several mRNA sequences that code for different proteins, okay? For eukaryotes, we can't do that. Why can we not do that? Yes?
Because you have on the 5 and you have the 1 CCAP.
Yeah.
So it's like an external factor and it's
Yeah, okay. Let me extend this for you a little bit. What on the prokaryotic mRNA makes it able to be polycistronic? Like instead of having a 5' cap to start translation, do you have a thought or should I give it to someone else?
I forgot what it stands for, but it's
Okay. Yeah, John, yeah.
The Shine-Dalgarno sequence?
Yes, there we go, the Shine-Dalgarno sequence, that's right. Do you know what the Shine-Dalgarno sequence is? Yes, man, you're on it, dude. Let's go. AGG. Okay, so you can put an AGG anywhere in that mRNA multiple times, right? So you could have AGG, then open reading frame, right? And again, remember the small subunit, the 16S subunit of the ribosome is what binds to the Shine-Dalgarno sequence and the AUG, right? That start codon. Then from there, everything can assemble on top. You move to the EP and A sites, right? With our amino acid tRNAs coming in, our peptidyl transferase creating those bonds, okay? And then later on, you have a stop codon. Then you can put another Shine-Dalgarno sequence and do it all over again on the same strand of mRNA. Okay, what's a compensation mechanism for this? I heard it said in the audience. What's a compensation mechanism for this? What could you do instead of making a bunch of proteins from one mRNA? What could you do instead? Yes? Yeah, I'm looking for something that like ties in a little bit more directly. Yes?
Polyproteins and breaking them up after translation.
Yeah, post-translational modifications, right? You can just cut them up, right? So instead of just making a bunch of separate proteins, which you could just make a really long polypeptide and then just cut it up and it's the same thing, right? It's the same thing. Okay, this is exactly what I said. 16S subunit, 5' methylated cap. You know the Shine-Dalgarno sequence now, AGG. Now we're going to move on to something that's related to being polycistronic, specifically the lac operon, okay? The lac operon. Okay, this, like, I guarantee you will be on the test. Like, hands down, I'm not a TA, but I'm telling you this is going to be on the test, okay? What I need you to do, I've got a few problems with this. I might skip a few of them because we're getting close on time here. Try to solve this one, okay? Just try to figure out what's going on. This image is just to help you, but just look at the plasmid and chromosome assignments there. All right, guys, I'm going to give you an answer for this. We're going to do one more of these. I have an extra problem that if you go back into the slides that I'll post after this lecture, you can do another practice one, okay? I think these are, yeah, I actually pulled these from our tutorial, so, you know, you should be familiar. Okay, so this is, I think, actually the hardest one, so this is the one to pay attention to. First thing to recognize, okay, we're looking at permease and beta-galactosidase, okay? You need to know the physiological implications of both of these. Beta-galactosidase is what breaks down lactose into allolactase, right? And that is what allows the repressor to then be let loose off of the operator, right? And so then our transcribing machinery can go through, okay? So let's talk about permease first, okay? So we see that a promoter on that plasmid is broken, okay? Which means nothing can be transcribed ever. Like, it's just broken. It's, you know, nothing's happening there. On the chromosome, you can see that the Y, that is the permease, is not functional. That is, it does not produce permease. So in no way ever is this meroploid going to produce, merodiploid, going to produce permease. What does that mean then? That means that in no way can lactose ever get into this cell, into this bacteria, right? So we don't have a feedback loop then. That's the idea. And so there's nothing that, like this functional beta-galactosidase now, the Z plus right there, right? There's no way of reinforcing that by getting lactose into the cell, breaking it down into allolactase, and then removing that like repressor. There's no way of doing that. And so that one is only expressed basally, okay? So permease, not produced at all. Beta-galactosidase, produced at a basal level. Only basal level. Okay, that's what I would say is the hardest example from our tutorial. Frankly, I'm going to skip the next two. So we have 15. This is another example. Yes?
What's the situation where it would be below basal levels?
Below basal levels. Okay, let me explain this. So you've got basal level, right, which is a little bit of production. The repressor is leaky, which means that mostly it stops transcription, but not 100%. There's always some getting through. Why is this the case physiologically? Because if some lactose is in the environment, right, you want to be able to get that into your cell. So you need to have some permease out there to allow it in, and you need to have some beta-galactosidase out there in the cell to break it down into allolactose, to then bind to the repressor, remove the repressor from the operator, and then allow more transcription, so to reinforce the feedback loop. That's the basal level, okay? But in this case, because our promoter doesn't function and our permease gene on the other, on the chromosome also doesn't function, then you can't even produce that basal level. Like there's no leakiness is happening, like nothing can be produced. There can be a basal level of the beta-galactosidase because it is functional, right there. It's a functional promoter, and so even with a repressor there, it can still do a basal level, okay? But that has no way of being reinforced because no lactose can get into the cell in the first place, right? So there's no way that I can then break it down. Yeah?
Even since the repressor on the chromosome, since it's broken, it still maintains a basal level though, even though it can't just continuously run?
So the repressor here, remember, there's a difference between acting in cis and acting in trans, right? Acting in cis is being DNA and working on DNA that's adjacent to where you are, right? So the operator is acting in cis, right? It's a portion of DNA that's acting on the promoter right next to it, right? But the repressor, I believe that's the term for it, is not acting in cis. It's acting in trans, right? So it produces a protein, and that protein is what then binds to the operator. So it doesn't matter if this one is not functional. We have another functional copy over on this plasmid. So the repressors from this plasmid, because again, they're in the same cell, are going to move over to the chromosomal DNA and be able to repress there. Yes?
So from there, what if we had a mutation, it was exactly the same, but we had a mutation where the operator was a C?
Yes. That means nothing can bind to the operator.
But then, even though Y is still negative, that lactose can still be like.
The problem there is that the promoter doesn't work. Remember, it goes operator promoter. So even if my operator can never bind a repressor, right? So long as nothing can ever bind to my promoter, I'm not going to get any transcription at all. Like, it's just not possible. So the promoter is the key thing. If you have a non-functional promoter, nothing can happen, right? My mic fell off. Does that make sense? This is something to think about. It's something to sit down and think about, okay? I have other examples in these slides, but they're just directly taken from your tutorial book. So if you want to look at your tutorial book, that's a great place to supplement your learning on this. But again, that was, I think, the most difficult one for me to conceptualize myself. So that's why I'm sharing it with you. Okay, so those were the other examples you can take a look at. What are two ways that transcription activators in eukaryotes, right, increase the rate of transcription in eukaryotes again? What mechanism? What do they do? Okay, this is something that either you remember or you don't. Frankly, if I hadn't looked over the slides today, I wouldn't have remembered this. This is like more recent stuff, but still kind of in the past. So this is actually the sweet spot that I would say, like for your studying, when you go home, right? Like pull out this slide set. Really take a look at ways of regulating eukaryotic and prokaryotic transcription. This is huge. Okay, so there's two different ways of how this works, right? You'll remember that we have enhancer regions on the DNA. This is a region of the DNA that activators and I believe repressors is the other term, can bind to, right? Okay, so here are the two ways that this works. First of all, you can bind basal factors, okay? So what happens is the activator binds to the enhancer, you create a loop, and there's mention of the actual mechanism by which the loop is made at the end of that slideshow that this comes from. So you make the loop, and then the activator, it may recruit a co-activator, it may not, right? It will bind the basal factors. What are the key basal factors, right? The TATA binding protein, that binds to the TATA box, 30 base pairs back from the +1 site. Distinguished from prokaryotes, which is 35 base pairs back and 10 base pairs back. Again, A's and T's at these sites, right? Binding the basal factors and then making it easier for transcription to occur. Instead of these basal factors having to float in, right? They're actually attracted by the activator. It's facilitated by the activator. Okay, the other way that this works is by looking at how the promoter is actually structured. Remember, DNA doesn't just sit around as chromatin all the time, right? Well, actually it does, but like I'm talking about a single like DNA with no proteins, right? It's not just a strand of DNA. It's wrapped around histones, right? And so the tightness of how these are wrapped corresponds to the ability to transcribe it, right? If these are tightly wrapped together, it's really hard for the basal factors to bind to the promoter and then work, right? Because you have to get rid of all these histones. But what our activator and co-activator are able to do is work on the promoter to get rid of some of those histones. So we go to euchromatin. Heterochromatin is where you have a bunch of histones in it. Euchromatin is where you get rid of those histones. So when you get rid of the histones, then the DNA itself is more accessible to the basal factors to then allow for transcription, okay? There are, again, repressors as well. That is something for you to take a look at. And I believe some things about hormones in there as well, so something to look at there too. Okay, another key thing here, on the activators themselves, it's question 18, on the activators, what two motifs are commonly found in the DNA binding regions of the activators? And what differentiates these two, okay? So the activator has to have some way of binding to the enhancer. What are the two common ways in which it binds to the enhancer? And what are those structures that support that binding? 30 seconds. What did you say?
No, it's wrong. Yeah, no, it's wrong already.
What? Okay, guys, we're gonna answer this. You remember it or you don't, okay? Zinc fingers, leucine zippers. There you go. Zinc fingers, because there's zinc chilling there, right? And fingers, because the attraction of the zinc, the charged zinc ion, creates bonding. So then you have these finger structures, right? Leucine zippers, because they're dimers and they're like zippers, okay? You can have the homodimer or the heterodimer, okay? And I believe there's some more significance to that. Frankly, I haven't studied that yet, so I'm gonna look into this more, but like, what's up with the homo and the heterodimer? I'd say take a look at that, right? And you can see how they actually fit into the DNA, right? Both of these have ways of inserting themselves into our helical DNA structure, okay? They fit in well. Okay, another thing to just remember. What are the assumptions for the Hardy-Weinberg equilibrium? Just say it. The difference? Look at that yourself. Yeah. 5 points, guys. 5 points. Think of them. What are they? Okay, we're gonna bring it back in. Okay, does someone want to volunteer what they think? Yes.
So the assumptions are every individual have the same chance for mating.
Yep. So random mating.
No migration from this population to other, and no migration from other to this population.
Yep, that's another point.
No
Good.
mutation.
No mutations. That's right.
A large number.
Yep. Like infinite number.
Yeah.
Yeah.
Yeah. True. And that's what.
Yeah. Okay. So like that's not listed right there. The population is infinite in size. The locus of interest, that's just kind of a specific way of putting it, has no effect on mating. That is, mating is completely random, okay? There are no new mutations in the population, so we're not having any of the mutations that we discussed in our slideshows, right? Substitution, deletion, translocation, etc., etc. There's no migration, of course, we covered that, and natural selection does not occur. Okay. Now, what I would have done, but like again, I see we're running out of time here, is to get you to run a Hardy-Weinberg equilibrium and then calculate the chi-square. But we're not going to do that. Okay? What I'm going to do is I'm going to post these slides and I'll post that as well. Okay. One last thing we're going to talk about. This is something you can look at yourself. PCR. Okay. Last thing, guys. Please stay with me here. We'll quiet down. Okay. This is going to take 1 minute. Okay. PCR, how does PCR work? I'm just going to walk through this here. Why do we need PCR? Can someone tell me why on earth would I want to run a PCR? Like, what does it do for me? Yes? It makes a lot of copies of a specific sequence of DNA that we're interested in. Why would I want to amplify a specific sequence? DNA sequencing. Yes, Sanger sequencing. That's right. What's another common application that we all relied on for the past few years? Yes. COVID tests. That's right. There's a bunch of other ones too. But this is the key idea. So we want to multiply a specific sequence. Now, we need to know what that sequence is, right? Not completely, but at the beginning. And I guess at the end as well, right? Primers. We need to make primers. So here's what we're going to do. We've got this DNA that we're interested in, okay? And it could be a whole fragment, but we know that It's somewhere on this large fragment that we need to copy, right? And we go to a website on our laptop, right? And we search on our laptop, please have like this sequence of primer. Then we place our order. It costs us $5. I asked about this in class. And then they ship it to you through like Amazon Prime or FedEx or whatever. And then you get it and you chuck it into a PCR machine, okay? What else do you add to that PCR machine? You got our primers. You've also got to add, man, this is taking so long, dNTPs, okay? What you're trying to do is replicate the DNA. So this is DNA replication, but in vitro, not in vivo, right? So we add our dNTPs so we can polymerize. What else do we need? We need DNA polymerase. Okay, great. We also need buffer to do this in. Okay, what is the process? We've got to denature. We got to move them apart, right? We got to split them apart so we can access both strands. We've got to anneal. So we've got to get the primers onto either strand, right? Then we got to extend. Okay, we extend those primers with DNA polymerase 2, not 3, not 1, DNA polymerase 2. Okay, so we do that. Hooray. Man, I borrowed these slides from our teacher. I'm regretting this. It takes so long. Okay, there we go. Boom. All right, that's our first round of PCR. Wow, a bazillion more to go. Okay, so we just keep doing this, right? And doing this over and over again. Now, I want you to notice the first time you run the PCR, you start with the primer, right? But then you keep going for the whole length of the fragment. There's no way of stopping. It goes for the whole length of fragment that you put in there. So not just the DNA of interest. It goes from the primer at the start of the DNA of interest all the way to the end of the fragment. On the second round, the primer is able to bind to the other side of that section of interest and then synthesize in the other way. So then you get fragments that are all the correct length. Okay, and they just keep popping up. There we are. Okay. All right. And that's what you get, all right? Okay, this is the end of the lecture. So first, I just want to say thank you all for coming all year. This is crazy. But thank you. Thank you. Yes. Some people dream about like going to the NHL. Okay, this is literally my dream. Like teaching lectures. So thank you so much. I want to wish you all the best on our exam. Study well tomorrow. And thank you all. You're wonderful classmates. Love you guys.